Take a look at the following pictures. One is the sky, and the other is a fog. What do these beautiful scenes have in common? These views are created by light scattering in the presence of tiny water molecules around the air. Likewise, scattering is a physical process where moving particles or waves are forced to deviate from a straight trajectory by localized non-uniformities in the medium. Scattering can occur in various forms, and physicists want to understand the scattering in a fundamental manner. The scattering problem can largely be categorized into two subsets. The first one is straightforward. It aims to compute the scattered field when an incident beam and a scatterer are given. This is called the Ford problem. The second one is the reverse mode of the Ford problem. It aims to find the structure of an unknown scatterer based on the measured scatter field. This is called the inverse problem. Solving both problems is important, but solving the inverse problem has more significances in fundamental physics and practical applications. It means that you can determine unknown 3D structures just by measuring the scattered waves or particles. This will surely remind you of many practical examples, including X-ray CT, electron microscopy, and 3D optical microscopy, where various important 3D structures should be inspected and investigated. If solving the inverse problem is so important, what is the challenge to do so? Mathematically, the preliminary step of solving the inverse problem is to fundamentally understand the governing scattering equation. This is called the Riemann-Schwinger equation. It intuitively describes how scattering waves continually encounter a scattering obstacle and propagate as a form of spherical waves. So this can also be interpreted as the Huygens principle. The problem of solving the problem is that wave scattering occurs multiple times according to the equation. In other words, multiple scattering always occurs. Understanding this multiple scattering has mind-boggled many scientists, and previous methods of tackling this multiple scattering have been either inaccurate or inefficient. Here are some previous studies of solving the multiple scattering problems. Some methods, including the Born approximation or the multi-slice method, fail to accurately describe the multiple scattering process. Other methods, including the FDTD, have been established as accurate ways of describing the multiple scattering process, but they were shown to be computationally inefficient. Consequently, when scientists computed multiple scattering in the inverse problem, they had to use efficient inversion algorithms at the huge expense of the inaccuracy in reconstructing the 3D unknown structures of scatterers. Because accurate and efficient inversion of multiple scattering is important in many research fields, our team has long studied how to deal with the inverse scattering problem. Then one day we found one of the most important papers published in 2016, and we were inspired to apply this to solving our problem. Moosung Lee and Hervé Hugonnet, under the guidance of Professor YongKeun Park, collaborated together to obtain positive results. Here is the essential principle described in the 2016 paper on how to efficiently and accurately compute the Riemann-Schwinger equation. For simplicity, we first discretize the equation in the following vectorized form. Then the equation can easily be solved simply by taking the matrix inversion as follows. However, inverting the matrix is practically beyond the current limits of the memory capacity, so we have to rely on alternative methods. One simplest way is to use the Taylor expansion to make the matrix a polynomial series. In physics, this is called the Born series. The problem of the Born series is that it diverges generally, simply because its value is often out of the radius of convergence. To resolve the divergence issue, the authors of the paper presented a concept of modifying the series terms so that the series can always converge. In this way, the Convergent Born series always converges and accurately and efficiently solves the fourth problem. Here we compare the Convergent Born series with FDTD, one of the well-established methods for computing the fourth problem. We simulated the misscattering by a 3-micron diameter silica bead in water and compared the simulation results with the theory. The result indicated that the Convergent Born series was more accurate and efficient than FDTD. We then applied the Convergent Born series immediately to solve the inverse problem. To do so, we defined an error function as the absolute sum of the squares of the differences between the estimated field and the measured field, and reduced the error by taking the gradient descent. This gradient descent theoretically required two sequences of using a forward solver, which we did so using the Convergent Born series. To make a rough analogy, we solved the inverse problem by backpropagating the error between the actual and the predicted scattered field and recovering the unknown scattering structure using the Convergent Born series. This is like a scene in Tenet, where the time is inverted and backpropagated to find the original shape of a crashed car. So here are the results. We tested the reconstruction of a spherical particle using various inverse solvers. As a result, the inverse solver using the Convergent Born series provided significantly improved reconstruction performance at the expense of affordably longer computation time. We extended the comparisons to variable samples, including multiple colloid microparticles and a live mouse red blood cell in simulations and experiments. All the results indicated that our proposed methods showed the best reconstruction performances. Here are the 3D galleries of experimental data of microparticles and the red blood cell that we measured. In addition to these small particles, we also successfully reconstructed larger RG cells and human pancreas tissues whose thickness far exceeded 10 micron. In these cases, our methods could improve the reconstruction fidelity and visualize the finer details inside the biological samples. Lastly, we also confirmed the repeatability of the reconstruction performance by improving the tomographic video of a genetically engineered T cell attacking a cancer cell. These results imply that the immediate applications of the present method can improve the fidelity of our quantitative biological analysis by circumventing the multiple scattering issues. Please check out the following article for the relevant contents. To recap, we have exploited the Convergent Born series, recently one of the most accurate and efficient forward solvers, in order to solve the inverse multiple scattering problem. We expect that this can be generally extended to various applications where understanding multiple scattering is fundamentally important. For more information, please refer to our recently published article.